Hello, welcome to the session about Ataxia Research Explained, Fruit Fly Models. My name is Dr. Celeste Seward. It is my pleasure to welcome you on behalf of the National Ataxia Foundation to this session. We get a lot of questions here at NAF about research models, what they mean, what they do, what information can this give us about ataxia research. This is part of this series we're hosting to shed a little bit more insight in how, well, all the amazing animal models that help us better understand ataxia. And with that, it is my absolute pleasure to introduce our speaker today, Dr. Sokol Todi. Dr. Todi is a professor in the Department of Pharmacology and Neurology at Wayne State University School of Medicine and the chair of the Department of Pharmacology. Dr. Todi's laboratory investigates mechanisms of neurodegeneration and neuroprotection in age-dependent neurodegenerative diseases caused by misfolded proteins, with a particular focus on polyglutamine disease proteins. His laboratory has invested in SCA3 for over a decade and has recently expanded to other SCAs, including SCA2, SCA7, and SCA17. Dr. Todi's laboratory uses cell culture and the fruit fly, Drosophila melanogaster models, to model SCA and other diseases to find potential therapeutic entry points that include small molecules as well as lifestyle options, such as exercise. Dr. Todi, thank you so much for joining us today. The floor is yours.
So let's thank you very, very much for having me here today. It's my pleasure to tell you about the little fly that can. And I will spend a fair amount of time with all kinds of details, but I will not test anybody at the end of the talk on any of them. So please feel free to forget them if you don't find them interesting. I've been using fruit flies actually since 2000 to conduct various types of research early on in my PhD work to study how flies hear and how that can teach us about diseases in humans that lead to deafness. And then later on as a postdoc, I moved into polyglutamine degeneration and ataxias, and that's what I've been doing in my lab for quite some time. Today, I will spend a bit of time in telling you why the flies are so beneficial, and then ending with what the flies have informed us about different diseases in humans and the types of drugs that they have helped us in identify, in improving, and in having better targets in the clinic. So the fruit flies, I'm sure you've seen them flying around. I probably have a few flying around here because we have a fly kitchen not very far from my office, and they tend to migrate whenever they can. They will come in males and females. The male is smaller than the female, and if you're interested on how we tell them apart, well, besides the size, the males have these little brushes in the front legs called the sex combs, and also have a darker abdomen than the females. But why? Why use fruit flies for research in general, and especially for human diseases in particular? You may have heard this before, but it bears repeating. About 75% of known human disease-related genes have actual functional orthologs in flies, which allows us to conduct all kinds of modeling of the diseases in the fruit fly. Flies reproduce very, very quickly. Every 10 to 12 days, we go from an egg that has been laid as a fertilized egg into a full adult, which can live to about 100 days if you're careful with them, because we can change the rapid pace of development in the fly. We can really coordinate these developments to be even quicker. If we keep them at, say, 25 degrees Celsius, which is comfortable for us, they'll be 10 to 12 days. If we raise it to 30 degrees Celsius, it'll be as early as 8 days. But if we cool it down more like 15 degrees Celsius, they can take about a month. So we can coordinate their development very carefully as well. They are powerful genetic tools. We have been able as researchers, and here I stand truly on the shoulders of giants who have paved this pathway. We can conduct all kinds of interesting experiments in the fly because of its tremendously flexible genetics and the huge amount of tools and number of tools, rather, that we have amassed over decades. So we can express specific genes, including disease-causing genes in specific tissues for a specific amount of time, and only in some cells, but not ones surrounding it. So we can really interrogate very, very in very minute detail exactly how these disease genes are impacting one cell versus another in the fly. We have a fully characterized Drosophila genome. We have the entire sequence, so we can make very careful and targeted deletions, insertions, mutations to understand the functions of these genes that cause disease in humans. And they are rather low cost and easy maintenance. They are quite inexpensive to keep, require minimal space and resources compared to, say, vertebrate models. The conservation of key pathways, whose identification has led to a number of Nobel Prizes in physiology or medicine, is very important. So fundamental physiological, biological processes are conserved from flies all the way to humans, making it much easier for us to understand these diseases at this small model and then interrogate them in larger models like mice, like rats, like primates. They have very high reproductive rates. A single female can lay hundreds of eggs, which allows us for very powerful experiments statistically. They allow us to conduct in vivo modeling, where we can model neurodegeneration, cancer, metabolic disorders, and more than that, through tissue-specific gene expression or mutation. They are animals that allow for behavioral assays. Drosophila exhibit learning behavior. They will also show locomotion, sleep, and learning. They will follow rather locomotion, sleep, and learning paradigms. They will show anger and anxiety in some very specific cases that maybe we should take with a grain of salt sometimes. But they do allow us to recapitulate in the fly diseases like epilepsy, as well as ones like diabetes. So we can absolutely follow this in the fly. And studies in flies face many fewer ethical constraints than vertebrate models, which simplifies approval and execution of experiments. These are rather just 3 photos of how we store flies. These are not from my lab, but you can see that we can maintain them in endless trays with fly vials. Each vial can contain hundreds of flies or thousands of them if we use bottles, and we can actually change them into new food every 3 to 4 weeks to continue growing them. And that's really all it takes. In fact, there is no other way to maintain flies. You have to propagate them from vial to vial to vial. You cannot, as in some other animals, freeze them. To resurrect them when you need them, you have to maintain them. And so, in fact, there are some flies that my lab has that have been around since the 1920s. And I'm very proud to maintain some of those lines for others to use when the need arises. The fruit fly cycle is very rapid, as I said, 10 to 12 days. We go from an embryo to a larva to a pupa, or what you would see as a fly in a cocoon. Once the flies are eclose or emerge from their pupal case, they'll live about 100 days. But the fact that we understand so well the developmental pathways of the fly allows us to also conduct studies that impact development in humans, especially for neurodevelopment, to understand how different mutations cause different diseases. I'll next give you a brief history of fly research. It was in the early 1900s that Drosophila was proposed as a model organism for genetic studies by Charles Woodworth. Thomas Hunt Morgan, the father of fly work, essentially, then with his many researchers who followed in his stead, conducted tremendous cutting-edge work in the 1910s to discover sex-linked inheritance. His lab was phenomenally important in foundational genetics because it was Morgan's lab that established the fly as a premier model for classical genetics. And thanks to him, we learned that a gene is linked to a chromosome for the first time. He won the Nobel Prize for that. In the 30s and 50s, a lot of work was conducted to map the genome to find out where different genes were located in the fly genome, and we'll take a very quick look at that in a few slides. Detailed genetic maps were constructed, laying the groundwork for functional genomics later on that have ultimately led us to understanding different diseases in the human condition. In the 1960s and 70s, a tremendous amount of work was done in understanding developmental genetics in the fly, and a lot of research on embryogenesis and patterning genes was fundamental in linking different pathways from flies all the way to humans in terms of their evolutionary conservation. In 1983, genes are shown to be conserved across species, linking fly genetics to vertebrate development. In 2000, the entire sequence of Drosophila melanogaster was completed. The full genome was published, and it became a foundational reference for comparative genomics in, well, now decades to come. And starting in the 1990s, disease modeling and functional genomics started taking place and expanding in the fly, with the fly being used to model human diseases and to screen for drug targets. The last portion of my talk will actually list the kinds of drugs that have been impacted by fly-based work. Several notable Nobel prizes were given for fly work, starting in 1933 with Thomas Hunt Morgan for discovering that genes are located on the chromosome using Drosophila, establishing the chromosomal theory of inheritance that applies to all of us. 1946, Hermann Müller demonstrated that X-rays induce mutations in the fly, and we actually have used this sort of X-ray induced mutations for quite some time in fly genetics to understand the role of different genes in any aspect of fly development and longevity. In 1995, Edward Lewis, Christian Nüsslein-Volhard, and Eric Wischhaus received a shared Nobel Prize in physiology or medicine for identifying genes controlling early embryonic development and body segmentation in the flies and revealing conserved developmental pathways through phyla. 2011, Bruce Beutler and Jules Hoffman were awarded the prize for discovering the role of the Toll gene in innate immunity in Drosophila, further uncovering conserved immune signaling mechanisms, and we'll see later on how these discoveries have led to various drugs targeting cancers in humans. And most recently, Jeff Hall, Michal Rosbach, and Michael Young received a Nobel Prize for their identification and elucidation of mechanisms for circadian rhythms using Drosophila as a model, and further confirming evolutionary conservation from flies all the way to us. Some of the similarities that keep emerging when we conduct fly work and further convince us that these are a fantastic model to understand biological processes, whether they are unfolding normally or linked to pathology, are conservations of genetic conservation, molecular pathways, disease mechanisms, cell responses, and behavioral phenotypes. So from the bottom there, flies can model aspects of locomotion, memory, sleep, and seizure-like activity in humans. Stress responses, apoptosis, autophagy, etc., are conserved, as are mechanisms like neurodegeneration, metabolism, cancer, and immunity. And we can model all of this in the fly. Specific pathways like signaling cascades in Notch and MAPK operate similarly. And you'll see these names pop up again when we talk about, or when I tell you rather, about various drugs that have been influenced by fly work. And of course, many core genes and their functions are shared. Now, unlike us, we have 23 of chromosomes in duplicate. There will be 46. Flies only have 4. And we start with chromosome number 2, which is this one right here. That's one outside, and that's the other duplicate. This contains a high density of genes involved in various aspects of neurodevelopment, signaling pathways, metabolism, and more. We have the third chromosome, or chromosome 3, which is important for genes related to embryogenesis, patterning, and cytoskeletal structure. Of course, we have the sex chromosomes, X, which you can also sometimes call chromosome 1, but I would not advise that. I would call it X. This is, of course, involves genes that are important for vital development and cell functions. And we have the Y chromosome, which encodes male fertility factor, but mostly it is a non-coding gene. We also have a very small chromosome that we call the dot, which is largely dispensable. You can get rid of a single one of the dots, and one single copy will be perfectly fine for flies. This only contains about 80 genes, mostly plays a role in chromatin regulation and gene silencing, essentially how the other genes are responding. In flies, having two X's, just like in humans, means that the fly is female. An X and a Y, just like with us, is a male. However, in case you're interested, it is not the Y chromosome that determines sex in the flies, it's the number of X's. And so an XXY will still be female. If you're interested, we can talk about that in the Q&A. We have lots of tools that fly biologists have been preparing over the last 110 years or so. And we have lots of collections that we all can tap into when we need to study something new. So we have expression systems that allow us to express something when we want it, where we want it, for as long as we want it, and even some very interesting systems where if you have 100 genes that comprise a specific tissue, you can select to express something or remove it only in a subset, so you can see how the brothers and sisters surrounding it of these cells are going to respond to whatever you have done to a subset of the cells. So we have a tremendous number of tools that we can play with, including, for example, having a female fly with a male brain or vice versa to understand how the brain can impact the morphology, locomotion, seizure aspects, whatever you want of an otherwise female fly and vice versa. We have access to available reagent collections. If I need a fruit fly line that I don't have in the lab, I can log on to a repository, and just a few days later, I can have it delivered to me for a very low cost. And this means that we, as fly people, collaborate very tightly together and help each other expand our researches. How would you use fruit flies? Well, we use fruit flies in many different ways. The primary tissues that we look at in an adult are going to be the fly brain, the fly eye. I'll show you some examples of how we use them in a moment. The alimentary system, so the gut and intestine in the fly, which you can see here how we have aspects in the fly that mirror the stomach, the kidneys, the large and the small intestine, and of course, we also will use the reproductive organs of the fly to model different diseases. Other things that we use that aren't featured here are the fat body, which is a thin layer of tissue underneath the cuticle of the fly that essentially functions as a combination of immune system, metabolic system, and digestive system in the fly. We'll use a heart tube. There isn't really a heart. There is a heart tube with multiple little hearts that push the endolymph around in the fly. And of course, we have conducted, as fly biologists, not me personally in this case, we have used our immune system to understand immune diseases in humans. Various diseases that have been studied in the fly are listed here, and they, of course, will include neurodegeneration like Parkinson's, amyotrophic lateral sclerosis, Huntington's, Alzheimer's, SCAs, etc. In terms of repeat expansion ataxias, SCAs 1 and 3 and others have been modeled consistently in the fly. We've done so for metabolic and endocrine disorders, muscle diseases, cancer and tumor biology, hematopoietic and leukemia models, epilepsy and seizure disorders, cardiac and channelopathy issues, and infections and immune disorders by looking at different diseases and by modeling different features in the fly. So, for example, for Parkinson's, how dopaminergic neurons are being lost. For repeat expansion ataxias, behavioral and locomotion decline over time. In metabolic diseases, endoplasmic reticulum stress. In terms of muscle diseases, muscle dysfunction, flight defects, shortened lifespan, etc. And so we are able to conduct these studies quite rapidly and in very large numbers and with minimal cost in the fly. The ataxias that have been modeled in the fly, the ones that we have studied are ones that for which we know a genetic cause. So SCA 1, 2, 3, 6, 7, and 17 that we know are inherited and whose genes we understand where the mutations are, these have been modeled in the fly. And they've been examined in the brain, in the eye, in the muscle, and in the case of FRDA, Friedreich ataxia, also in the heart tube of the fly. We have used mostly genetic models for this, but in some cases, like with FRDA, we have also used drug-induced ataxia by using oxidative stressors that will lead to issues with motility as well as muscle damage and issues with longevity in the fly. What do we look for when we model diseases in the fruit flies? Well, it depends on where we are expressing the protein or gene of interest. Usually, we'll do this in the brain and in neurons, and we'll look at neurons, eye, and also glia. So look for things like loss of neurons, brain atrophy, synaptic defects, protein aggregation, and behavioral decline, whether this is climbing, they can't climb as well, or memory, that they don't remember exactly where the reward is, for example, or where the light is. Muscle pathology for diseases where we have clear muscular involvement, we will look at the structure of the muscle, whether the indirect flight muscle or the body wall muscle, and we'll look at muscle degeneration, impaired flight, reduced climbing activity, and at the cellular level, mitochondrial disruption, since we know the mitochondria are going to be tremendously important to the muscle and the amount of energy that they require. For cardiac function, we'll look at the heart tube, and we will look at things like reduced heart rate, morphological anomalies, lifespan reduction, arrhythmia, meaning the different tubes aren't functioning in synchrony. Metabolic dysregulation, we'll look at the fat body, gut and the muscle. We'll look at things such as altered lipid storage, oxidative stress, glucose handling defects, and mitochondrial dysfunction. We'll look for eye morphology at the retina photoreceptors with different kinds of the phenotype of the eye, which I'll show you in a moment. Other things we look at, so let's say you have immune activation, we'll look at cytokines, immune gene induction, and altered hemocyte number and behavior. For developmental defects, reproductive defects, and behavioral changes, all of this, again, we'll look at tissues that are corresponding to these diseases. And I see that there's a question in the Q&A, so maybe I can actually address it right now. This is a good moment. So
sure.
Larry is asking, is there a way that fruit fly models can predict or explain why repeat expansion get longer or shorter from one generation to another, such as in SCA27B? Larry, that is a fantastic question. And actually, I'll tell you that I have a new postdoc in my lab who is starting to investigate this very question. The one thing I will give you preemptively as a caveat is that not all repeat expansion disorders will show expansion across generations in the fly. Some do and some don't. So for the ones that don't, we can't quite get to that question, but for the ones that do show it, we will be able to have some clues why that happens. And so please stay tuned. Hopefully in the next year or so, we'll have some details ready for the community to look at. I hope that helps, and I hope I understood your question properly. So a lot of the things that we do in the fly have to do with the fly eye. So here is a perfectly normal fly eye, nice color. You see this sort of pseudopupil dark spot in the center that will disappear as the eyes are malformed. So you'll see in an eye that is not formed properly or is showing degeneration or thinning of the eye, that dark spot is gone, and you have some variegation in eye color from red to orange to yellow. As things get even worse, the eye gets even thinner, you'll see indentations in the eye, and when it's really, 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 really bad, you'll see an eye like this that you can barely call an eye anymore. And so we can actually assign numerical values to the different type of degeneration that we see, and we can use that for all kinds of screens in the fly to see whether a specific gene is being helpful or not in a condition, and how different drugs may help, or in some cases hurt, when we're modeling a disease in the fly. Other things we look at are longevity. Don't worry about these colors, but these would be in black. A normal fly would live about 100 days. You'll see how expression of different disease genes will shorten that tremendously by a third or less overall. And these are studies that can be done in a blinded way, meaning the people who are monitoring the flies don't know exactly what genotype the fly is. And so there is a lot of bias removed from that assessment when they're counting flies, and also looking at their motility, which I can show you right here. We have a few, these are all male flies, by the way. No, I cannot tell from this angle, but I just know that they are. Females don't move as quickly. There is that. But you saw a hand that slapped them down, and you'll see that hand appear again. And we can take these measurements to see how many flies go to the top in, say, 5, 15, or 30 seconds, or how many flies are at which stage or at which length on the vial in 3 seconds. And we can conduct a lot of assays that way. And those assays are especially important when we do something like exercising flies. And yes, you can. Flies move against gravity. And so if you design a machine that repeatedly drops them down, the flies will move against gravity, and they'll go up and up and up and up. And you can do this for 3-4 weeks, and they will actually show improved endurance to long-term training. So you can see as the machine drops them, the flies are jumping up. You'll see it again right here. Whereas these ones here have their siblings that are not being allowed to be exercised. So you can have tens of thousands of flies either exercised or not at the same time, and understand why, if it does so, why exercise will be making things better, or in some cases worse. And we can design exactly how gentle that drop is, how rapid it is, and how long they are allowed to rest between drops to mimic different types of exercise for different types of ataxias or other diseases. So how have flies informed other research? Well, we'll take a few examples here in the next few slides. In Parkinson's disease, a key protein that's involved in Parkinson's is alpha-synuclein. The work that was done in the flies, it helped to identify the role of this protein in terms of its toxicity for dopaminergic neurons, and it formed drug screening targets, and it helped with research in mice and in zebrafish. So by doing this work in flies, we're able to then go more rapidly into mice and zebrafish with very specific questions, by eliminating the broader questions by doing work in flies. This also applies to Huntington's disease and FRDA, where studies in the fly have revealed the importance of polyglutamine toxicity and how different genes may exacerbate or alleviate it, supporting development of potential therapeutic compounds in Huntington's, and also informing us on the role of antioxidants and metabolic therapeutics development for FRDA. We've done the same thing for Alzheimer's, ALS, and SCA3 by modeling them and by understanding the role of key specific proteins, whether A-beta and tau in Alzheimer's, TDP43 and FUS in ALS, or Ataxin-3 in SCA3, to help us understand what potential mechanisms we can target for therapeutics, but also what mechanisms in the fly, so endogenous mechanisms in the cells and in neurons and in glia, are being impacted by the disease proteins that we are expressing. A lot of work has been conducted with cancer, diabetes, and cardiomyopathies. In a list of drugs that I'll bring up in the next few slides, you'll see that quite a number of cancer-related drugs benefited from work in the fly. And so it was this work, so whether EGFR or RAS or Notch pathways conducted in the fly, that allowed for work in mice and in human xenograft models to understand cooperative oncogene interactions and tumor suppressor networks. And so these supported personalized drug screening in fly avatars for specific mutations that have been found in different patients, that have led, again, to drug design and improvement. The same thing was done for diabetes and cardiomyopathies, where work in the flies has helped to understand us better at a minute level, the genetics, the molecular pathways, and the cellular components involved for various genes that are involved in diabetes and issues with cardiomyopathies. And so this brings me to the end, essentially, step by step of this presentation. Next, I have a number of drugs that I'm going to show you or briefly go through that have been developed or informed by fly-based research. A lot of them are ones that are important for metastatic and non-metastatic cancers overall. So we have things like vismodegib, sonidegib, glasdegib, nirogacestat, and imiquimod that are important for different tumors and cancers, and how the fly work has helped. So the hedgehog pathways have been critical in understanding the role of smoothened as a protein in advanced metastatic and basal cell carcinoma, adults specifically, and vismodegib was designed based on this information. The same applies to sonidegib and to some extent glasdegib overall, and how they have been important. And you can see that they started in the 90s, but has accelerated a bit more in the 2000s after the full publication of the Drosophila genome. Additional ones are here: trametinib, cobimetinib, binimetinib, selumetinib, and imatinib, all benefiting from various types of work done in the fly overall, and again, targeting different types of melanomas overall. But we also have a number of drugs that are used in patients that benefit neurological disorders. So it's not just the cancers that have benefited from this work, it's also diseases that neurologists encounter in their practices. And this will include epilepsy, focal seizures, bipolar disorder. These are going to be especially important because a lot of the epilepsy drugs and epilepsy studies were based originally on a study of the shaker mutation in the fly, where it's a fly line where it has a specific mutation that if you bang it, meaning if you just tap it on the table, the fly will start shaking instead of moving up. And that led to discovery of very important ion channels that then could be targeted for therapeutics further down the line. Additional ones are listed here that have to, again, do with motility issues, including some in Huntington's chorea and tardive dyskinesia, in Alzheimer's disease, and neurological manifestations, different models of the fly that are used here to identify potential drug targets and to refine their use for specific mutations in humans. And that brings me to the end of this conversation. We have lots of time for questions. And so I already see a couple of questions here from Amanda. Do you anticipate your work will be impacted by the current federal funding issues for your lab and others? And the answer is yes, I do. I do see it impacted directly and indirectly. Directly because of various assertions from the current administration that there has to be a very high focus on human-based research. And while that is a wonderful goal to have, that doesn't mean that we can move into the type of research in the way that they may be anticipating as non-researchers themselves. And so we still need the kind of work that we're doing and that my colleagues are doing with vertebrate animals, especially in mice and in rats, to understand the minutiae of these diseases before we can move on to, say, drug trial of some sort or another. Yes, we may be able to use things like iPSCs or cell models or organoids in our labs to understand these diseases or potential therapeutic entry points by using a more human-like model. However, I will caution here that when we are deriving cells from one version to another, say from a fibroblast to a neuron, what we're also doing is we are scraping off completely some of the programming that is necessary to understand the disease initiation and progression. So I am a bigger fan of using all the different models in the best way possible to inform each other from one step to the other. How do you tell if a fly is angry? They show aggression, actually. Males will show aggression when it comes to mating. And there are some great studies that have been conducted and are being conducted by people like Ori Schaefer, Maria de la Paz Fernandez, who are now at the University of Indiana, Bloomington, who study how different times of the day and how that impacts the behavioral of the flies and their aggression towards other males when there's a female around for competition, because ultimately it's always a competition that leads them to decide, the female that is, on to which male she will be receptive for copulation, and in which order as well, for that matter. So that's very important. Do you actually draw blood from the flies to do testing? We don't. Flies don't have blood. They have hemolymph. We can use it to understand certain to do some metabolic studies, say when certain metabolites are up or down regulated, when we express a gene of interest, but we don't tend to draw it. We just, for lack of a better word, squish the entire fly and use the entire mass to understand what's happening in that case. That being said, there are beautiful studies we can do by visualizing how things move through the hemolymph of the fly by having fluorescent proteins, and you can see them being popped around with the fluorescent microscope in a live fly. It is a gorgeous view.
Well, thank you so much, Dr. Todi. As a reminder for folks to please put your questions in the Q&A window. We'll get through as much as we can in the next 20 minutes or so.
And I'm also happy to go over some slides that maybe I went through very quickly if people have specific questions.
100%. While we're waiting for folks to type in the chat, just a follow-up question from me. You talked a little bit about how sex is determined in fruit flies and something about two X chromosomes with a Y chromosome still being female. Could you please walk us through that a little bit more detail?
So yeah, it's the ratio of X's. One of the X's will be inactivated, not completely, partly, but it is not the Y chromosome. So in us, in humans, the Y chromosome determines the sex of the developing fetus because of the SRY gene that will ultimately determine which organs are going to fade away and which ones are going to be used to become male organs. And I can give you a whole human physiology reproductive session if you want me to do that. I'm actually teaching it again later this semester. So that's easy. But in flies, that's not how it works. It's the axis. And it's even more interesting. Every single cell has its own sex. So you can determine, that's how you can make a fly that has a male brain but female body that will respond differently to, say, locomotion and endurance training than the vice versa, a male fly with a female brain. It's a brain that dictates how it's going to respond. And so as a ratio of the X's, then the only purpose for the Y gene is to allow the expression of some genes to be used for copulation and successful, essentially, mating. Not really required so much for the rest. So when I said you can have an XXY fly, that is perfectly true. However, they're sterile. So there is enough of a incongruency in the system that while they will come out and they will perform like females and they'll be receptive to males, they will be sterile flies in the end.
That is fascinating. Another thing you talked about,
biology.
Just biology is fascinating. It's marvels what nature can do. You also talked a little bit when you were outlining the different types of ataxia models that are fruit flies, the difference between genetic and drug-induced models. Could you talk us a little bit more about what those means and how perhaps they're used in Friedreich's ataxia?
So unfortunately, we only have the Friedreich's ataxia that has been used by using oxidative stress to sort of induce ataxia. We don't really have other models that I am aware of that do that that have really done that. I suppose you could use flies to model overall issues with mobility by giving flies different drugs. And we've done that for the purposes of drug testing. So let's say we have a compound that one of our colleagues has designed to prevent the aggregation of a specific protein, let's say an A-beta-42 for Alzheimer's disease in this case. Does the feeding of this compound to the fly lead to locomotor issues in the first place? And yes, it can. And yes, it does. And maybe we could use that as a way to model issues of mobility, but we use it more for the purposes of safety. The vast, vast, vast majority of the models in the fly are going to be, or have been so far, transgenic, where we express a gene that we do know causes issues in humans and is a cause for, or a cause for a specific disease. And then we express it in different tissues and we see what happens. If we express, say, the SCA17 gene in muscle versus in neurons versus in glia, what can we understand about the importance of this gene in each of these tissues? And the reason why I think that's important is because while we think of ataxias and other mobility diseases largely being neuro or neuromuscular, other tissues do also appear to be impacted, perhaps in a different level, perhaps to a different extent. But that's one thing that, at least in my lab, more recently, we have started to try and understand is how other non-neuronal or muscular tissues are impacted in different diseases, including in ataxias. And that's my very long answer to your question. And I don't even know if I actually answered it, but you
No,
can
you
keep probing.
totally did. Thank you for the clarification. On that topic, though, because you're now talking about tissue-specific differences in flies, and they are so small. How do you isolate different types of cells from different aspects of the fly accurately, just because it is such a tiny organism?
It is tiny, absolutely. And if you think that a fly is tiny, think about the antenna of the fly and the second antennal segment of the fly, which is what I used to dissect with scissors to study as a PhD student. So yeah, with dissection microscopes, with high magnification, and with very steady hands, we will dissect brains, cut them in half to use one portion for one versus another. It's a lot of maybe seemingly tedious work, but it's worth it. And we do it, and we have listened to a lot of podcasts while we're doing it.
I'm just trying to imagine you now dissecting all these brains while listening to a
Oh yeah,
podcast in the background.
it's
Sounds like
great.
a lot of work to get your data.
It's
Speaking
worth it.
speaking of your data, you mentioned a little bit about the exercise tests that you are doing with your fruit flies. Can you tell us a little bit more about that? Can you test out different kinds of exercise in fruit fly models? And if that's the
Absolutely.
case, how do you do that?
So we can do high impact versus low impact exercises, high frequency versus low frequency exercises. So how quickly they're dropped, how much of a distance they're dropped, how much of a time they have between drops. We can adjust all of that, as well as how soft their landing is. And we've done that, or rather, I should say now, it's one of the newest faculty members in my department, former postdoc of mine, Dr. Alison Swikovsky, who's doing this work with different kinds of neuromuscular diseases, including Kennedy's disease and SCAs. And she's finding that depending on the type of exercise, endurance training, for example, has been beneficial for SCA2, but not so much for SCA3 and detrimental for SBMA. Thank you, Amanda. And so now she's trying to determine what kind of exercise can be beneficial to one type of ataxia versus another, and how those pathways may be changed in one disease versus another. And it's interesting in that in work that she's doing in the flies and work that a collaborator of hers is doing in Huntington's patients with exercise, they're finding similar things about the type of intervention that is helpful versus beneficial to humans and flies, and preparing a manuscript together, paralleling these differences so they can publish it. But it's very interesting how informative this can be. And to give you one more example, the genetic screen in the flies, not done by me, but done by Dr. Swikovsky with her collaborators, found a gene in the fly that's the same gene in mice. And if you target the same gene in mice, it has the same beneficial effect as if the mice were exercising overall. So huge conservation from flies to mice, even at that level.
Well, we were talking about how nature can be fascinating, but science can be fascinating too. These are very exciting developments. Best wishes to everyone involved trying to do this work.
Thank you.
I'm not seeing any other questions in the chat, so we'll end with one last question from me then. It's a lot of people are using fruit fly models for all sorts of neurodegenerative research and for all sorts of ataxia research. What are you most excited about for research that is happening right now and maybe happening over the next few years in terms of progress that can be made or developments that can be, well, developed?
For me, what's extremely exciting is beginning to understand what pathways, what cell processes, what components at the genetic and epigenetic level these different diseases share and where they diverge from each other. And so I'm very curious. So there is that component. The other one is the tissue connection, how tissues talk to one another, whether a disease protein is expressed on them or not. And so understanding the impact of one tissue to another and understanding the cross-reference of these different diseases to understand whether a similar intervention can be used across all of them, or it should only be used in this subset of diseases, but not the other. I think that's becoming clearer and clearer, the current models that we have, and I'm very excited about it.
Well, thank you so much, Dr. Todi, for joining us today and telling us more about the little flies that can, Drosophila melanogaster, and how the amazing work that's being done in order to better understand ataxia. We really appreciate your
Thank
time
you
and
for
your
having me.
expertise.
Thank you
And
for
to
having
all,
me. Thanks, everybody. Yeah.
and to all of you who attended for the past hour or so, we really appreciate you spending this time with us. I hope you learned something new about fruit flies and how they can impact ataxia research. Wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care, folks.
Thank you.